Hello, and welcome back to Psych 300, Brain and Behavior. We are beginning our Chapter 4, or the neuron, and we will be considering several topics within the neuron. First, we'll talk about some early work. Let me just make sure that we're capturing everything here. Yep, looks good. We will be talking about some early work, looking at the communication within the neuron. We'll also be talking about how the neuron is much like a battery, and it has a resting potential, has the ability to do something. And then we'll talk about how when that energy is tapped into, it generates an action potential. And then we'll talk about the conduction of that change in charge across the membrane. And then we'll talk about postsynaptic potentials. But first, I'd like to bring to your attention, because we're starting to talk about some chemicals and some electricity, and your book has two really good resources. On page 88 and 89, there is a basic review of chemistry that does a good job of talking about sodium and potassium, chloride ions, and how there are these charged molecules. So you might want to review that to try to see when I'm talking about the balance of the charges across the membrane. That might help provide a little bit more context. On pages 112, and there is a review of the basics of electricity and electrical stimulation, so to give you a little bit more background about that. So use those resources and you'll have a better understanding of this material that we'll be covering. I do also want to point out that some of the stuff in this chapter is going to be a little bit tougher than what we've talked about already. So I'm going to probably say things a couple of times. That'll give you the indication that I really want you to stop, pause, take notes, re-listen to that section to make sure you really understand the resting potential, how that's generated, and then what happens to the ion channels, which we'll talk about during the action potential. So that means I may be going a little bit slower and I may be a little bit more repetitive, but I think this is some of the hardest topics to get through in this class, is the neural communication, or communication within the neuron. So let's give a little context, background, historical kind of view on how the central nervous system communicates. And we're really going to be starting with Galvani. He was the first person who really suggested maybe communication in the central nervous system has an electrical component. Before this, people really, in terms of arguing about how the central nervous system functions, thought their best guess was that there are animal spirits. And I kind of alluded to that when we were talking about the ventricles, that people argue that the ventricles were filled with animal spirits, and that's what caused people to be animated or to move or engage in behavior. So here what we see is that Galvani was the first to suggest maybe it's not animal spirits, maybe it's another type of phenomenon we can study, such as electricity. And there's a little bit of folklore around Galvani, how it came to this conclusion, and I'll share the story with you. It's definitely in historical neuroscience textbooks, so there may be some documented evidence for this. The story goes that he was walking through an open-air market, kind of like a farmer's market, and at this time there was a storm on the horizon. It was rolling in, and you could see lightning off in the distance. And as he was walking through there, he noticed that there were these frog legs. People during this time must have sold frog legs and had them freshly prepared. And they were hanging from brass clips. And as he was walking through, he noticed that the frog legs were actually twitching. So both seeing the storm rolling in, the electricity generated from the lightning, and seeing that these frog legs were twitching in response to these brass clips, he wondered if he applied a charge to frog legs, if he applied a little electricity, would he be able to generate movement? Would that be the basis? And what he found is by going, and you can kind of see some of the apparatus that he used here, where there was a wheel that he had some magnets, and he was able to develop a charge by spinning the magnets and then apply that charge to different components, and he was actually able to generate movements in the leg of a frog, suggesting maybe electricity was an important part of the communication system within the central nervous system. So that's where we get a transition from animal spirits to actually observable kind of phenomena. It wasn't really until the work of Hodgkin and Huxley that we had a better idea of how the central nervous system uses this electricity as a communication system. And it was their work with an axon from a squid. Now these squid have these very large axons, and this makes ideal preparation for studying the electrical activity in the central nervous system, because it's so big, it's easy to isolate that axon and then study it in a dish. So I think one part of their breakthrough to understanding the electrical activity in the central nervous system comes from the fact that they found the appropriate animal model, and a model that involved a very large axon that they could isolate and then keep alive in a dish. Another part of their breakthrough came from a device that allowed them to record the very, very small, minuscule charge that was present in the squid axon. And that involved the this device here, which basically is an oscilloscope. And an oscilloscope, what it does is it can detect very small charges on the range of 70 to 100 millivolts. These are very small charges. We couldn't power a light with these kinds of charges. And so what they do is that once you connect a squid axon to some wires, and there is this beam of electrons that are constantly hitting the screen. If you close that circuit, what happens is the very small charge that's present there will cause that beam of electrons to move just ever so much. And so now you can actually measure these changes in the electrical activity, and see that the axon from the squid actually is kind of like a battery where it stores energy. And that energy that it stores is enough to move just one electron that's being pulsed, electrons being pulsed through that area. So this work won them the Nobel Prize in 1963, discovering an appropriate animal model and using the appropriate kind of tool to measure these small charges across the membrane. And what they found is that in a resting state, so you take this part of the squid's axon out, keep it alive in a dish, and you record from it, it's got a potential or a voltage difference across the membrane of 70 millivolts. So that is a small amount, but it's a very consistent amount. Now this is where it gets interesting, I find. If you look across, this is an invertebrate, the squid is an invertebrate. If we look across other animals such as rats, non-human primates, birds, reptiles that have any kind of nervous system, ganglion, even earthworms, what we find is that their resting potential is very, very close to this negative 70 millivolts. They might be a little bit lower, a little bit higher, but we see that they're all very close to that. That shows that there is, like we talked about in our origins chapter, there is this continuity, where there's this similarity across different organisms, and at this level, the charge that's resting there, ready to do something, is at a very similar level across these different species. So the question became, how do these neurons maintain this charge? What is serving the function? What are the forces acting to engage this charge? And there are two forces that I'd like to talk about first to give you some understanding of how they work. First is diffusion, and diffusion is pretty straightforward. You can imagine maybe you've made Kool-Aid in the past, and if you just plop the packet into one spot, into a jar or a jug of water, what you'll see is that it'll come to one location where you put it in, and then gradually over time it'll start to move from an area of higher concentration to an area of lower concentration. So if you weren't to stir it and you just waited for enough time and you came back, what you find is that the Kool-Aid would be completely dissolved throughout the jar of water. And that's the process of diffusion, where compounds or elements, molecules want to move from an area of higher concentration to an area of lower concentration. This is something that we see throughout many mediums, and this is an important force that will contribute to generating that resting potential. Another important force is electrostatic pressure. Electrostatic pressure basically involves what we know about magnets. If you put two similar poles together, they repel. Two opposite poles together, and they attract. So if you were to use a magnet and try to push that magnet like these two mice here have together, and the poles were aligned, so north was getting close to north, they would push against each other. However, if you were to switch it, so north was going towards south, they would attract each other. What we see is that if you have two positively charged ions, they will repel. But if you have a positively charged ion and a negatively charged ion, they attract. So opposites attract. That's the idea behind electrostatic pressure in terms of electricity. Likes repel, opposites attract. So with these two forces in mind, what we're going to do is set up a stage and try to indicate all the critical players that are going to be on this stage, and have the situation where both diffusion and electrostatic pressure are going to be the plot points to tell this story. So let's first try to set the stage. And our stage is here at the cell membrane, the phospholipid bilayer. Remember, those tails like to be away from water, whereas the heads like to be next to water, and so they arrange in this fashion here. So what we see is this is our phospholipid bilayer, and there's this extracellular space up here. So this is outside of the neuron, and intracellular space here. This is inside of the neuron. And what we'll see, first of all, is we have a large number of negatively charged protein ions within the neuron. So just as a basis here, we see that we have a lot of negatively charged protein ions here. So that kind of sets the stage that there's a lot of negative ions inside of the neuron. Now, across this membrane, we can distribute or have several different charged ions we'll talk about. And the first is potassium. Potassium, or K+ here, this yellow block, is showing that it's at highest concentration inside of the neuron, and it's positively charged, K+. So the box indicates that there is a lot of concentration inside of the neuron. So because of that concentration and the way diffusion works, potassium wants to leave. So it wants to leave the neuron. It wants to go from an area of higher concentration to an area of lower concentration. And it's positively charged, so if we look at electrostatic, we see that in general it's negative charge inside of the neuron, and so opposites, what do? They attract. So potassium wants to stay within the neuron. So you have a diffusion wanting potassium to move out, and electrostatic pressure where it wants to stay in. So potassium, under these resting conditions, is balanced, and so it wants to stay where it's at. There's no pressure acting on it. Diffusion is balancing out the electrostatic pressure. Now, another ion that's distributed across the cell membrane are chloride ions. And chloride ions are in larger concentration on the outside of the neuron. So chloride ions are much greater numbers on the outside of the neuron, and so that means through diffusion it wants to move in. But chloride ions are negatively charged, and so that means that when you think about electrostatic pressure, chloride ions are negative, and the large number of protein ions here inside of the neuron are negatively charged. So electrostatic means that it wants to stay out. It's repelled. Like potassium is balanced. So under these resting potential conditions, we see that potassium is balanced, chloride is balanced. Now, we have one last player here we'll talk about, one last actor, and that is sodium, Na+. Sodium is in its highest concentration outside of the neuron, and it also is positively charged. So both through diffusion and electrostatic pressure, sodium wants to move within the neuron. It wants to move inside because it has its highest concentration outside, and so it wants to move to an area of lower concentration, which is inside of the neuron. It also is positively charged. Remember, we have all those protein ions in here that make, in general, the inside of the neuron negative. So both through diffusion and electrostatic pressure, sodium wants to move into the neuron. So if we look at all of them again, what we see is potassium is balanced, electrostatic pressure wants it to come in, diffusion wants it to come out, chloride ions are balanced, diffusion wants to move in, electrostatic wants it to move out, and sodium is our only ion across the membrane where its highest concentrations on the outside and electrostatic pressure are pushing it inside. So both of those things are pushing in the same direction or moving into the neuron. Now, one thing to do to keep in mind, try to figure out how do I keep track of all these ions, where they're located. Well, if you remember that there are just three that we're really interested in, and think of these neurons bathing in salt water, what is sodium chloride? That's right, it's salt. So you can kind of think of them in this fluid out here. The neuron, outside the neuron, is salt water. And think of sodium chloride as being the salt that goes in that water. And that should help you remember that it's in its highest concentration outside, whereas potassium has its highest concentration inside. So that's a little thing for you to kind of keep in mind as we're going through this. So because of this differential influence of both diffusion as well as electrostatic pressure, sodium is really eager to enter into the neuron, but because the phospholipid bilayer doesn't just let these readily pass through, we have to then provide some entrance to this. But before talking about that, we'll also say that sometimes there is a tiny bit of slippage. And so in order to keep sodium outside that might slip through and potassium that may slip in, we have a sodium-potassium pump that's always working to make sure that sodium is a higher concentration outside and potassium is a higher concentration inside. There's just an occasional slippage that can happen. And then we'll see that this is helping to maintain this concentration gradient. So it's kind of like, I don't know if maybe your home, you have a sump pump, and what happens is water starts to fill up and this pump will move it out of the house. Well, what this pump does is it kicks out any sodium that it can grasp onto, and it brings in any potassium it can bring into. So the sodium-potassium pump is always chugging, trying to keep that, if there's any kind of leakage anywhere through the membrane. So it's trying to deal with that. Now, that maintains our charge across the membrane at that negative 78 millivolts. How do we start to generate a action potential or a change in that? How do we tap into that energy? And there are a couple of important a couple of important features that we have, and these are voltage sensitive channels. And when we talked about these in our last chapter, and some of the things that are being made within the neuron, were activation of the DNA, and we can make these proteins. One of the things we talked about were channels, and what we can have are voltage sensitive channels. So here, what happens is if there's a change in the voltage of the membrane, we somehow are able to get away from that 70 millivolts up to around 50 millivolts, and we'll talk a little bit more about that number. So if we can get from 70 up to 50 millivolts, all of a sudden a channel will open. And the channels are kind of like little doors in this membrane that does a pretty good job of keeping sodium out and potassium in. But if we can get up to negative 50 millivolts, then we can start to change the function of open a door in that membrane and allow certain ions to move through that membrane. So voltage-sensitive channels are found in certain locations across the neuron, and we'll see where they're located and how they function. So again, we make these as little doors that we can keep closed or not allow anything, but if there's a certain change in the membrane potential, such as we can get away from 70 millivolts, negative 70 millivolts, up to negative 50 millivolts, all of a sudden these doors will open up and they'll let those ions pass through that are want to pass through. So we have the stage set. We know that we have some ion channels that are selective to potassium, some are selective to sodium, and we have the sodium-potassium pump always working very hard to keep that balance of sodium outside of the neuron and potassium in higher concentrations inside of the neuron, and we're at that negative 70 millivolts. You'll notice that here we have negative 50 millivolts, here we have negative 50 millivolts, and negative 70 millivolts. This is where we're recording from that axon. Here we have our potassium channel that is sensitive to voltage of the membrane, and we have a sodium channel. So typically potassium goes through here, typically sodium goes through here, and we have our sodium-potassium pump chugging away here, trying to keep that balance. Now, if we were to apply a little electrical charge, so let's say we just added 10 across the membrane here, we get up to 60. Nothing would happen. If we added a little bit more and we got to maybe negative 55, nothing would happen. What we see is that there's a threshold. If we can get the charge across the membrane, above negative 50 millivolts, all of a sudden we generate what's referred to as an action potential. We'll talk and characterize this a little bit more, but all of a sudden things start to change dramatically, and we unleash that energy, and we see that we set off a whole cascade of events, the sequence of events. So when we get above that negative 50 millivolts, the first thing that happens is that the sodium channels start to open. Sodium channels open. And what do we know about sodium? Sodium wants to move into the neuron, right? Because both through diffusion, its highest concentration is outside the neuron, and electrostatic pressure, opposites attract. Sodium's positive and these protein ions are negative. It wants to move in, rush into the neuron. So as we're adding all those positively charged sodium ions into the neuron, inside of the neuron, we see that now we get past 50, it starts to rocket up. It actually goes above 0 and still continues to climb. So opening those sodium channels are important for depolarization, or changing that polarization back to 0, or starting to go above a little bit. So that's depolarization of phase of an action potential, because these sodium channels open. Now, as the sodium channels open, what happens as it starts to turn positive? So inside of the neuron, because we're adding all these sodium ions, what happens is the inside of the neuron starts to turn positive. And that has a influence here on potassium. Remember, while at the resting potential, potassium was balanced, right? It was in its highest concentration inside of the neuron, but it's positively charged and inside the neuron is negatively charged. So both through diffusion and electrostatic pressure, potassium wants to stay inside. But we're actually changing the inside of the neuron to where it now is starting to become positively charged. So that when we open up those voltage-sensitive potassium channels, they start to open up. Sodium's rushing in. These potassium channels open up. Now potassium is in the highest concentration inside the neuron. And it's also inside the neuron is positively charged. So now potassium wants to move outside of the neuron. And so right after those sodium channels open, potassium channels start to open. And that's what slows this change in the membrane potential. So you see this is starting to grow, but it also is so it starts here at negative 70 millivolts. We're able to get it above negative 50. Those sodium channels open, and this skyrockets. It's just going up faster and faster, but then it starts to slow down, and that's because the potassium channels are starting to open. As those potassium channels open, we start to now allow potassium to flow out. And as those potassium channels start to flow out, what happens is our sodium channels close, preventing any more sodium from rushing into the neuron. So sodium can't come into the neuron, and what happens is those potassium channels continue to stay open, and potassium continues to rush out of the neuron, subtracting a positive charge from inside of the neuron. And because we're subtracting all this positive charge, now we go back down below zero. Potassium is continuing to rush out. Finally, those potassium channels will close, but look where we wind up. We wind up down here around 100 millivolts. And this is the hyperpolarization phase. So we've gone through a couple of phases here. First, we have the resting potential. Then we have depolarization. During depolarization, sodium channels open up. Repolarization has our potassium channels opening up and our sodium channels starting to close. And then during hyperpolarization, where we go below that negative 70 millivolts, our potassium channels now close, and we're down here at negative 100 millivolts, and that quickly returns back to, after those potassium channels close, back to the threshold of negative 70 millivolts. And all throughout this, we have the sodium-potassium pump chugging away, trying to kick out those sodium ions, positively charged ions, and bringing in those potassium-charged ions. So that's all the time going on, trying to maintain that balance. And so this is a single action potential that we see here. It starts with the resting potential, and then it ends with the resting potential. We have depolarization, repolarization, hyperpolarization, and then resting potential. Those are the phases of it. Now what I want to do for you is to go through this again, of how this changes across how the channels and the concentrations and the electrostatic pressure changes across the action potential. We'll see why this is important when we move on to looking at other features of the action potential here. So let's go back here through the resting potential. And remember that we have diffusion and electrostatic pressures functioning on these ions across the membrane. Really, chloride isn't playing a big role here in this specific example that we're going through. Our major players are both potassium and sodium, and of course the sodium-potassium pump, and all these negative ion proteins that are inside of the neuron. So at the resting potential, we're right at negative 70 millivolts. If we're able to get above negative 50, all of a sudden those sodium channels open up. And remember, sodium is on the outside of the neuron, and it's positively charged. So sodium's mostly on the outside, so diffusion, it wants to move in. Electrostatic, because it's positively charged. Opposites attract. It wants to move in to the neuron. It wants to move inside. And so when we get above that negative 50 millivolts, sodium channels open up, and sodium rushes in to the neuron. Now, as the sodium rushes in, it's adding a positive charge inside the neuron, and it will continue to do that and grow above that. This is the depolarization phase. Now, as that's happening, we have some potassium channels that start to open up, and it slows that ascent. It slows it down, and those potassium channels, because it's positively charged, potassium is in the highest concentration inside the neuron. We're adding all that sodium, and inside the neuron is becoming positive. Potassium now wants to move out of the neuron, and as we add that positive charge inside the neuron, and these potassium channels are now open, potassium starts to rush out of the neuron, and because of that, we start to see a dramatic drop or repolarization of the membrane. So it starts to dramatically drop. As it's dropping, we see that the sodium channels close. So those sodium channels close, preventing any sodium from leaving or entering the neuron as it starts to go back down to this negative polarity across the membrane. So potassium is rushing out, and it continues to rush out, and in fact, it does so well that it gets down below negative 70 millivolts, and we're down here at around 100. We're down at around 100 millivolts, and that's hyperpolarization. After those potassium channels finally close, what happens is the neuron will return back to the resting potential very quickly, and now we're at again, the resting potential are negative 70 millivolts. So this should be negative 70 millivolts. We're back at that where we started the resting potential. And this is an action potential here. And we can see that there's an exchange or a change in the membrane potential that occurs and depends on these voltage-sensitive channels, the function of diffusion, as well as electrostatic pressure, and that the adding the charge. And this is a really important thing to understand, that we have to add some electricity to this to get it above that negative 50 millivolts. In the next chapter, we'll talk about how getting this from negative 70 to above negative 50 involves a chemical reaction between neurons. So we'll talk about that, how that works. But in the lab, what we'll do is just apply a little charge to the membrane potential. So I think this is a good point to pause or maybe rewind and go through this to make sure you understand the sequence of ion channels opening. Remember, sodium opens, potassium opens, sodium closes, potassium closes, and the dynamic nature of how the membrane potential changes from negative 70, it goes all the way up to positive 30, and then it goes all the way down to negative 100. These are all dependent on diffusion and electrostatic pressure, as well as these voltage-gated ion channels and the sodium-potassium pump trying to keep that diffusion function in the same direction. Now, one thing I want to contextualize for you, that we're talking about a lot of chemistry. We're talking about electricity, and you're like, why are we doing this? This is a brain and behavior class. Why is this important? And this electrical signal is within the neuron. It's so important for the functioning of the central nervous system. And one of the things we know is that these are chemical electrical reactions that are occurring. And if you change pressure, so if we were to look at how fast things react at ground level, sea level, versus in the mountains, we know that they proceed at different rates. That's why when you're baking a cake, you need to adjust your baking times because things are different at a higher elevation than if you're at sea level. And so that reflects different pressure that you have to take into account for the chemical reactions that are necessary for baking or other processes. But it's not just unique to baking, it's all chemical reactions. If you change the pressure, you're going to change the rate of these chemical reactions. And so this helps explain what happens when somebody loses consciousness after having a closed head injury. That there's this, just like we said about the skull, it's a closed box. And when you get a mosquito bite, you see swelling. Well, if you have a significant head injury, or your closed head injury, and your brain is going to start to swell, that increase in pressure is going to change this rate of this chemical reaction, and it's not going to function, and you won't be able to communicate information down the neuron. You won't have this change or generation of an action potential that will then communicate information within the neuron. So that's why this is so important to understand. As we're going through and generating these action potentials, that's part of the communication of the central nervous system. And if you change the pressure within the central nervous system, then you won't have an ability to generate these action potentials, and then you'll lose consciousness. So that's a real important point to tie in. Why do we do this? We'll also see that there are certain chemicals that can change how these channels function, and may also change or alter states of consciousness. And we'll talk about those in a little bit in our synapse chapter, but that's what we'll build up into for our drugs and behavior chapter, how drugs have their effect, and some of them can work here as well. So it's a really important foundational material to understand. Now let's talk a little bit more about the properties of the action potential. First, how quickly does the action potential occur? I mean, how quick can one of these things happen? The way we talk about it, it took me some time to go through these different steps. So how long does it take it to unfold? And you'll notice up here on the x-axis we have a 1 and a 2. So what are we talking about? Well, the action potential unfolds within 1 millisecond. Now, I'm not sure, maybe some of you are aware, but how many milliseconds are in a second? Does anybody know? There are 1,000 milliseconds in 1 second. So that means that we can generate in a neuron around 1,000 action potentials within 1 second. It's pretty amazing, the time scale here. So here we see a typical action potential being generated, and then we're varying the time scale. Here's 3 milliseconds. We have 2 being generated. Here is all the way out to 40 milliseconds, and we can see again here this generation of these action potentials. And what you notice, and what becomes clear across here, is that we have this range from -100 to +30 on the y-axis. That's the change in the voltage across the membrane. What we notice is that that really doesn't change all that much. And so that gets into our next characteristic, that it's each action potential is a discrete event. So if you generate an action potential here, you get above that -50, it generates an exact pattern. So it will always look like this. It will always have this characteristic. And you can see that here, these are all action potentials, and you've lost some of that resolution here because we had to compress the time scale so much. Instead of being from 0 to 3, we're now from 0 to 40. And so you can just see the positive and the negative end of that. But these each are individual action potentials. And they're not varying a great deal in the maximum or the minimum that we have. So each action potential is a discrete event. It's an all or none kind of phenomenon, much like flushing a toilet. You can flush a toilet and once, but you can't just flush it again. You have to wait for it to fill again. So there's this discrete event that you apply enough pressure, then you don't flush the toilet. So that begs the question of how do we code for rate, or how do we code for stimuli of varying intensity? And what we do is we'll code by rate. So we're not going to see an increase. So if we push our finger with a certain amount of pressure and then we push even harder, we can tell those differences in pressure. Or if we're looking at a very dim light or a very bright light, or we hear a very soft noise or a very loud noise, how do we code for that? Well, we code in terms of rate, how many times it fires, that neuron is firing, generating action potentials in a certain time frame. So here we see a very high rate of firing that's occurring. That's a very intense stimulus. Here we don't see any firing really. We see one action potential, but here there isn't much going on. Maybe the stimulus isn't applied. And then we apply, you can imagine here, seeing a very bright light, and then it gradually fades away. This you could think of as maybe like a light bulb here flashing, and then it gradually fades away. And so you see that decrease in the firing rate as we get farther away from the light is becoming progressively more dim. So the rate is how we code for the intensity of the stimulus. Now, there's one caveat to this idea that action potentials are discrete events. And that is the fact that you can, with enough energy, actually generate another action potential within a millisecond, but it takes much more energy than just getting above negative 50 millivolts. It will require more stimulation, a little bit extra punch to do that. So here is our typical action potential, and what we see is that we can divide the action potential into an absolute refractory period, and the absolute refractory period is during depolarization and repolarization. However, during hyperpolarization, that's where it's down below negative 100 millivolts, it enters the relative refractory period. This is where if you put a lot of stimulation in during the relative refractory period, more than normal, then you can actually generate another action potential. So here, if you were to, normally, this is an action potential, you'd add a bit of energy, to get above negative 50 millivolts, you generate this action potential, and then you'd be ready, the same amount of energy would generate another action potential after here. But during hyperpolarization, if you really punch this neuron, increase the amount of energy, you're able to generate another action potential. So during that refractory period, you can actually generate another action potential, but you have to apply a lot more stimulation, a lot more change in the polarity of the membrane to get there. It's not impossible, but it's going to be harder than what happens if it was just during the resting potential. So that's something to keep in mind too, is that we can even get a little bit further down in terms of below a millisecond. But in general, what we see is that we can fire an action potential within a millisecond and be ready to go the very next millisecond to fire another action potential. But if we're having a lot of stimulation, then we can actually trigger another action potential during the hyperpolarization phase. So action potentials are discrete events. Either they occur or not, depending if they get above that negative 50 millivolts. We have rate coding, and we can kind of see if there's an even more intense stimulation, we can, during the hyperpolarization phase, actually trigger another action potential, but it's going to take a lot more energy than what it takes during just the resting potential. So we've covered a lot of ground here in terms of resting potential and action potential. And what I think you should do is give yourself a check to see, go through, make sure you understand a lot of these terms, how the sodium channels open up, how the potassium channels open up, and sodium channels close during each of those phases. Ask yourself, do you understand the time frame that an action potential occurs? Remember, you can generate an action potential within a millisecond, so you can have 1,000 action potentials within a second. And be able to talk about what is a relative refractory period and an absolute refractory period. These are all very important concepts. So we'll talk about another feature. So what, after you've generated an action potential, what happens? And that means we need to go back and look at our axon that we have recorded from in the membrane. Remember, this is the work that Hodgkin Huxley won the Nobel Prize for, recording from this. And what happens is that that membrane potential will actually start to spread out. So you have the sodium channels opening up and the potassium channels opening up, and you have this action potential, and it spreads, this change in the membrane potential spreads across the membrane, and it then will make it to the next sodium-potassium channel, opening that sodium and potassium channel and triggering another action potential. So it's conducted down the axon or across the membrane, and opening up the next kind of sodium-potassium channel. So here we see that membrane spreading, and it makes it just far enough to open up this sodium and potassium channel that re-energizes it, and then it will spread to the next portion where there's maybe another sodium-potassium channel. And so we can conduct it down the axon. Now, one of the things that we know about conducting electricity across wires is that the farther you send a signal along a wire, the lower that electricity is going to be, because it's going to lose energy to the environment. This is called cable properties. So if we had just a regular axon here going from the cell body, here's the cell body of a neuron, and we were able to generate a current here, and what we'd see is as we measure just this, and that we've stripped away all these other, sodium, potassium, we're just looking at what happens to that change in the membrane potential. What we'd see is that as we get farther away from that stimulation, it decreases in the magnitude because it's losing electricity to the environment. And it gets smaller and smaller to finally where it wouldn't be very strong at all, or it'd be completely gone. Think of I don't know if you, as a kid, you ever did this, but you took two strings and you tied two soup cans together or two cups together, and you can kind of talk at it and hear the other one. Well, at a short distance, it was easy to hear that, and then as you increase the distance, it was harder and harder to hear that. And somewhat the same thing happens in very old, a long time ago, when they'd have Morse code, where they had sent electrical signal. And as they got farther and farther away, it was harder and harder to hear that electricity because you was losing more. One of the things that they found was able to help this was covering the wire. So if they would cover the wire with some kind of insulator, they were able to see that they could send the signal far, way farther than if it wasn't covered. And that's what myelination does. Myelination helps to prevent that loss of that energy to the environment, so it protects it and allows that spread of the membrane potential much farther. So now the membrane potential is spreading much farther. And if we look at actually how this functions, what we see is that you have a full action potential here. It decreases in magnitude, then it's re-energized here at the node of Ranvier, then it spreads underneath this myelination provided here by the Schwann cell. Now here's a little question. What part of the nervous system are Schwann cells found in? That's right, in the peripheral nervous system. What is the cell that provides myelination in the central nervous system? That's right, oligodendroglial cells. So this myelination prevents that cable property or loss of energy to the environment. So as that membrane is spreading, it then re-energizes here at the node of Ranvier, and then it decreases and re-energizes. What's doing the regeneration or the increase in that? Well, we have sodium-potassium pumps located only at these nodes of Ranvier. Right here between where that myelination is located. So here we have a sodium-potassium pump opening, so we get a nice action potential. It gradually decreases in magnitude, but it stays above that negative 50 millivolts. So when it gets over here, when it spreads over here to this sodium-potassium pump, what happens is it's enough to open that sodium-potassium, re-energizing that action potential, and then it spreads over here to this next set of sodium-potassium pumps, re-energizing that, energizing that action potential again, and then it spreads across this membrane, and it'll dance, or what we refer to as saltatory conduction down the membrane. So we see that there's this re-energization of the membrane at each node of Ranvier, where here we see the maximum. It decreases down but stays above that negative 50 millivolts, our resting potential. It stays above that, so it's enough to open up the next sodium-potassium channel. Now, this brings us to a another aspect of this conduction that we can talk about, is the fact that we see that this is moving in a certain direction. And the question is, why does it move in this direction? Why, when it gets here, doesn't it go, oh, I'm going to go back this way? Why does it have a directionality in this conduction down the neuron? And the answer is that because we have the refractory period, there's a certain amount of time in which you can't fire another neuron. So as this is spreading across the neuron, it can't go back after it reaches here. It can't have the same power to go back and open up these channels. So once it starts in this direction, it's going to continue going in this direction, because that refractory period is basically stopping it from refiring again. So that refractory period is really important from the action potential getting halfway down the axon and saying, oh, I'm an action potential, I don't care what direction I'm going. Maybe I'll start going backwards towards the cell body. No, because we have a refractory period, we can't fire another action potential until we wait a certain amount of time. Then we can allow this to travel and in the same direction that we started out. So that's a real important detail here of the saltatory conduction, is that the refractory period that we talked about earlier here, that there's this certain amount of time that as this is coming down, we cannot trigger another action potential, prevents this from moving in the other direction, just deciding that it gets halfway down the axon and wants to change direction. No, the refractory period is a stopgap that prevents it from going backwards, and so we'll always get it moving in the correct direction because of that. So one other thing I like to bring up here is that we see how important the refractory period is and the spread of the membrane potential under the myelination. What do you think would happen if we removed some of the myelination? Well, what we would see would happen would be that we'd lose. So here's the sodium-potassium channels here, and we're able to trigger an action potential. It comes there, we re-energize it, and it starts to spread, but because it loses too much energy to the environment, it drops below 50%, that negative 50 millivolts. It's maybe negative 65 millivolts. And so that's not enough to open up that next sodium channel, and the action potential just stops right there. So removing that myelination is really problematic for conducting that electrical signal across the neuron. Do you know of any neurological disorder that involves removal or elimination of myelination in the peripheral or central nervous system? In the central nervous system, we see multiple sclerosis, or MS, is a disorder where there is a loss of myelination of those oligodendroglial cells. We see that there is this inability to communicate the signal. And it is a very challenging disorder for people because it can go into remission. It can occur in different locations in the brain. So you can see very different symptoms, from visual to auditory to attentional to motor to memory. And it reflects the fact that you're losing these myelination and these action potentials that normally would require that insulation to prevent it from losing too much energy so that it can energize that next sodium-potassium channel. But if you eliminate that myelin, now it loses too much energy to the environment, and it won't stay at that negative 50 millivolts, and that signal that's important for maybe memory or movement or sensory systems is lost. So it just will not be able to continue on down the axon, and that information will be lost. You won't be able to process that information. So that gives you another tie-in of why are we talking about saltatory conduction? Why are we talking about these nodes of Ranvier and where these sodium-potassium channels are located? Well, understanding this can help us develop treatment or understand the disorder so we can develop new treatments. And so that's why it's really important of how this molecular work translates to understanding these behavioral problems and how they're manifested. So our final topic that we'll talk about today are postsynaptic potentials. And this is work that John Eccles had conducted, and he won the Nobel Prize for this. This is really going to be, I think, a kind of a nice transition topic for our next lecture when we talk about the synapse and how the synapse works or functions, because we're getting into this idea, okay, we can stimulate one neuron and it changes the electrical activity in another neuron, but it depends on some kind of synapse, and we're going to talk about that next. But first, what do we mean by we stimulate this pathway and it changes the electrical activity or the potential in this neuron? Well, Eccles was stimulating certain neurons, and what he found is that it would gradually change the membrane potential. So it would show a gradual increase as he was stimulating this pathway, where it was getting closer and closer to being above that negative 50 millivolts. But if he stopped, then it would gradually kind of decay down. So this is a excitatory postsynaptic potential. Here, if he stimulated this pathway, he would get a EPSP, where we're getting close to that negative 50 millivolts, but not enough to generate an action potential. So he's stimulating this neuron A, or this pathway A, but he's actually recording from this motor neuron. And what he sees is that it's building, it's getting close, then he stops that, and then it just gradually decays down and isn't able to generate an action potential. If he stimulates another pathway, what he found was interesting, that stimulating this pathway but recording from the same motor neuron, that instead of seeing an increase, he could actually see where it starts out at negative 70 millivolts, but it starts to go down. It goes down to negative 100 millivolts. And then once he stops that stimulation, what he sees is that now it goes back to the baseline. So he could stimulate a neuron or a group of neurons, a pathway, and get a excitatory or very close to generating an action potential here, but not enough. And then if he stimulates a completely different pathway, what happens is he actually can inhibit the neuron or get it further away, like in that hyperpolarization phase. he can get it further away from being able to generate an action potential. And that further away from generating an action potential, really turning this motor neuron off, is called an inhibitory postsynaptic potential, or an IPSP. So he discovered that some pathways into a neuron can excite it, but other pathways can inhibit it. And what's really important for whether, there are two things that are really important for whether a neuron stimulating the excitatory or inhibitory pathways will generate a neuron, is first the timing or the spacing between when stimuli are given. Here you can give one stimulation and we see an EPSP being generated here. Remember, it has to get above that negative 50 millivolts to see an action potential. So stimulating this excitatory pathway, we see just one stimulation there and one stimulation there isn't really enough to generate an action potential. If we apply this same stimulation to the inhibitory pathway, we see that it gradually decreases and then it gradually decreases here. So it's not enough to generate an action potential. I mean, it's actually getting farther. It would be harder to generate an action potential here and here. We're getting closer, but not enough here. What happens if we then make the spacing between the stimuli, the electrical stimulation here and here, closer together in time? What we see is they start to summate. So here we see this is showing a growing, but if we apply the second stimulation very close in time from that excitatory pathway, now there's this additive effect. It's still not enough. It's still not enough to generate an action potential here, but it's way more than what we see with them spaced further apart in time. Here we see the same thing in inhibitory, where if they're close together, we see that it drives it down even farther. It would be even harder to generate an action potential here. What happens if we then do it even closer together? We see that we get even closer. So if we had a third stimulus applied right here, then we'd be able to see an action potential. So these are all EPSPs. These are all excitatory postsynaptic potentials, and if you put them closer together in time, then you'll be able to generate an action potential. These are all inhibitory postsynaptic potentials here, and if you put them close enough, you can completely shut down that neuron and prevent it from firing. So this illustrates that within the central nervous system, you have this battle between excitation and inhibition. So that's the major kind of dimensions that you have, excitation and inhibition, where if you have the excitation spaced very close together in time, you'll be able to generate an action potential. However, if you have a great deal number of inhibitory postsynaptic potentials close in time, you can completely shut down that neuron. So temporal summation is an important component, but spatial summation, where they occur on the neuron, is also very important. So here's where you can stimulate S1 and S2, these two excitatory pathways that are from two different areas, synapsing on different areas of the neuron. And what we see is they aren't enough to influence each other here, so you have that temporal summation, but because they're on two different locations, they don't have an effect. However, if you put them both on the same side here, now what we can see is that they have a much bigger effect than if they were farther apart. So not only do you have temporal summation, but where those neurons are coming from, the spatial location on that neuron is another important factor. So you can imagine that if you touch your skin very far apart or close together, with the exact same pressure, you're the closer together you are, you're going to see that there's this multiplicative function because they're close together in space, and you're going to be more likely to detect that than if they're too far apart. So the location where this pathway is coming into the neuron and synapsing is also important. So you have spatial summation, where it's coming in and synapsing. So if you have multiple synapses or connections between these two neurons that are close in space, you can generate an action potential. So putting this all together, this idea of postsynaptic potentials, we're getting information in from each neuron receives information from some cells that will be inhibitory and generate inhibitory postsynaptic potentials. They'll get some information or some contacts from neurons that are excitatory, and when those neurons fire, they'll produce excitatory postsynaptic potentials. And then when you summate across all of these, across the cell body, I kind of think of it like a checking account, where you can have some credits and debits. And here at the axon hillock, right where the axon leaves the cell body or the soma, there's that great summation where you have to see, what am I? Have I withdrawn too much, or do I have enough to do this, to make this purchase? If you have more temporal summation and spatial summation, then you'll be able to generate an action potential, down the axon. So here, what we see, that if there's enough excitatory postsynaptic potentials in a certain amount of time in a certain spatial location, and are able to outnumber all these inhibitory postsynaptic potentials, then you'll generate an action potential down the axon, and that will then continue that information. But however, if you have too many IPSPs or not enough EPSPs in a certain time frame or spatial location, then the axon hillock will say, no, we're not going to generate that action potential, and this neuron, that's where the signal will stop. So you have excitatory postsynaptic potentials and inhibitory postsynaptic potentials coming in from many different connections, from many different neurons. They converge and come to the cell body. As they generate across the cell body, they hit that axon hillock. The axon hillock is the major deciding point that will determine whether an action potential will be generated down the axon or not. So if you have more EPSPs, excitatory postsynaptic potentials, than IPSPs, and they occur within a quick enough temporal time frame, they occur close enough in time, and there's enough of them in a certain spatial location, when they reach the axon hillock, in this area right here on the axon, then you'll generate an action. But if there are too many IPSPs, or the EPSPs occur too slowly, or they're too distributed across the neuron, the axon hillock will say, nope, you didn't reach my threshold, and the axon will not fire an action potential and come to influence the next stage of processing. So the axon hillock of the neuron, that part where the axon leaves the cell body, is really important. It's the threshold. Is there enough excitation to warrant generating the action potential down this axon? So just to review this again, I want to go through what temporal and spatial summation is and talk about this idea that he can stimulate. Eccles stimulated one pathway and saw these EPSPs, or excitatory postsynaptic potentials. They were getting this neuron closer and closer to firing an action potential, getting above that negative 50 millivolts, but not enough. And you can also see stimulating another pathway generated IPSPs, or inhibitory postsynaptic potentials. And it was pushing that neuron farther and farther away from generating an action potential. So it would be harder with those. Now, what you see is that there are two important dimensions. Does the stimulation occur close in time? As you get closer into time, we see that those stimuli can be additive and get closer to that threshold. We can see also for our IPSPs, if it gets closer and closer in time, it's going to be harder and harder to generate an action potential. So it's going to be harder and harder to generate that action potential, and it will cancel out any EPSPs. But also, it depends on where that neuron is receiving the information. If it's from two different areas, you're not going to see that summation. But if you have it close together, those two neurons will summate, and they'll have a larger net effect than if they're farther away. So that is an example there, that there is closer in time and space can have a bigger effect than just closer in time alone. So both where that synapse is occurring and when that stimulation is occurring is very important. But it's always against the background of other IPSPs and EPSPs. And the axon hillock is that major deciding point that will determine whether or not that action potential is fired down the neuron. So this kind of concludes our discussion of communication within the neuron. This is half of the story. So we know that communication in the central nervous system is electrical, and that's what we part of it is electrical, and that's what we covered in this chapter. Communication with the neuron is electrical. The next chapter will talk about communication between neurons, which is chemical. So you can kind of think of neural system communication as being a combination of electrical and chemical signals. We talked about some early work that got us focused on electricity. We discussed how the resting potential is established by the distribution of ions across the membrane and electrostatic pressure, as well as diffusion. We talked about the different phases of the action potential. That was kind of tough. You want to review that material before you attempt the lecture quiz or the practice exams to make sure that you understand how that's functioning across there, across the different phases of the action potential and, the absolute and relative refractory period. Then we talked about conduction of the action potential down the axon and some of those important things. We saw the refractory period is important for determining the direction that it goes. And then we spent this time discussing the postsynaptic potentials. All right, thank you for joining me, and talk to you again in our next chapter.